You've probably heard a ton about editing our genes, rewriting the very blueprint of life. But have you ever stopped to wonder how that blueprint gets written in the first place? Well, today we're looking at both sides of that coin, how nature authors our genetic story and how we've figured out how to become its editors.
It's absolutely mind-blowing that we have the technology to edit DNA. But that incredible fact kind of jumps over a more fundamental question, doesn't it? Before we can even think about editing the book, we have to understand the author. So how does life actually write its own code?
To answer that, we've got to start at square one. Think of our genome as this enormous, incredibly complex book. And before you can write or edit a book, you first need to understand the letters, the alphabet it's written in.
And here's the thing. Making these genetic letters isn't some standalone job in the cell. As this quote gets at, it's this grand finale where all these different metabolic pathways we talk about, all the ways our cells make and use energy, all converge. It's like the central hub for the entire cellular factory.
So this fundamental letter is called a nucleotide, and it's really made of three distinct parts. You've got the base, that's the actual letter, your A, T, C, or G. Then there's a ribose sugar, which is kind of like the piece of paper the letter's written on. And finally, a phosphate group, which acts like a staple, letting us link all these letters together in a long chain.
Now, these letters fall into two basic families. You have the purines, that's adenine and guanine, and the pyrimidines, cytosine, thymine, and uracil. The big difference is their shape. Purines have this double ring structure, and the source material had this fantastic, kind of goofy way to remember it. The Pope is pure, and the purine looks like a Pope hat. You know what? It's silly, but it works.
Okay, so we've got our letters, we know what they look like. Now, how does the cellular factory actually, make them?
Well, our cells are super practical. They basically have two strategies. The first is called de novo synthesis, which literally means building from new. It's like cooking a gourmet meal totally from scratch. It's complex, it takes a ton of energy. The second option is the salvage pathway. This is just straight up recycling, grabbing spare parts from our food or from old DNA and RNA that's been broken down. It's way more efficient, and honestly, it's the cell's preferred method. That energy-guzzling from-scratch method, that's usually only for when the cell is growing so fast it can't recycle quickly enough.
And that from-scratch pathway is amazing. It's like this intricate assembly line. The cell pulls in raw materials from all over. Then, it uses these special molecules made from folate to add one single carbon atom at a time, meticulously building those ring shapes we saw before. Once the base is done, it slaps on the sugar and the phosphate groups, and there you have it, a brand new nucleotide ready to go.
So, that's the story of the author, right? The beautiful, complex process of how life writes its own code. But what happens when there's a typo in the book? That's where the editor comes in. And now we're going to shift gears and talk about CRISPR, the incredible tool that lets us go into that genetic text and make precise changes. CRISPR sounds like something straight out of a sci-fi movie, but its origins are actually ancient and bacterial. The name itself is a mouthful, clustered, regularly interspaced, short palindromic repeats. But all it really is, at its core, is a defense system that bacteria evolved to find and literally chop up the DNA of viruses that were trying to attack them. And scientists had the brilliant idea to harness that system for our own purposes.
When we use it, the CRISPR system really boils down to two key parts. First, you've got the guide RNA. You can just think of this as a molecular GPS. It's a short little sequence that we design in a lab to match a very specific spot in the genome. The second part is a Cas enzyme, like the famous Cas9. And that's easy. It's just a pair of molecular scissors. The GPS tells the scissors exactly where to go. So the whole process is beautiful in its simplicity. The Cas9 enzyme, holding onto its guide RNA, just zips along the DNA, scanning for a match. When it finds the target sequence, it does one last check for a tiny signal right next to it called a PAM. If everything lines up, the target and the PAM, the Cas9 scissors activate and make a clean cut right through both strands of the DNA. But here's where it gets really powerful. That initial cut, that's just the beginning. Scientists had taken that basic system and modified it, evolved it, and turned it into an entire toolbox of different kinds of editors, each one with its own special skill.
So everything starts with that cut, that double-strand break, or DSB. When this happens, the cell basically panics. It sounds an alarm and kicks its natural DNA repair machinery into high gear. And it's by hijacking those natural repair processes that scientists can actually introduce new edits into the code. The first big evolution of this tool was the nickase. By tweaking the Cas9 enzyme just a little bit, scientists made it so that it only cuts one strand of the DNA instead of both. It's like going from a big pair of scissors to a surgeon's scalpel. It's a much gentler approach that gives you a lot more precision and control.
Then things got even more precise with base editing. This is really clever. They use a dead Cas9 that can't cut at all. It just acts like a delivery drone, and attached to it is another enzyme that can do a bit of molecular alchemy, chemically changing one DNA letter directly into another, like turning a C into a T, without ever breaking the DNA's backbone. It's like using a pencil with an eraser to fix one letter, instead of cutting out the whole word.
And one of the most advanced tools we have now is prime editing. You can think of this as the search and replace function from a word processor, but for our genes. It uses that gentle nickase to snip one strand, but it also brings along its own template and an enzyme that writes new genetic information directly into that spot. It can insert letters, delete them, or swap any letter for any other. It is incredibly versatile.
And get this, it's not just one type of Cas enzyme. Scientists have found a whole family of them in different bacteria. SpeedCas9 is the classic, the one you hear about most. But there's also SACCas9, which is much smaller, making it easier to pack into viruses to deliver into cells. Then you have others, like Cas12a, that make different kinds of cuts. It's like having a whole workshop full of different scissors, each one perfect for a specific job.
So, we've gone on quite a journey. We've gone from understanding how life's book is written to seeing how we can now step in as editors. Let's finish up by looking at what this incredible new power really means for all of us.
Because this is why it all matters. The potential applications they're just staggering. We're talking about the real possibility of correcting the single-letter typos that cause devastating diseases like sickle cell or cystic fibrosis. We can engineer a person's own immune cells to hunt down and kill cancer. We can design crops that can survive drought and provide more nutrition. The possibilities touch literally every part of our world.
And that brings us to the final, and maybe the most important question. For the first time in the history of life on this planet, one species, us, has gained access to the source code. We're learning how it's written, and we're building the tools to rewrite it. The purely scientific questions are rapidly becoming profound societal ones. Now that we have this power, what kind of world will we choose to build? What stories will we decide to tell?